Morning, everyone. Thank you for attending. We're going to have some folks coming in a little bit late here. It won't cause too much of a disturbance. But today I would love to welcome y'all. And here they come back to our candidates seminar for our aquatic weed scientist position. It's my pleasure to introduce Eli Russell. Eli is originally from a very small town in Alabama known as Alabama. It's one of those places in the state you may have not been through. It's not exactly a destination, but as with a number of the brightest in the state of Alabama, he found his way to Auburn University, and where he got his bachelor's degree in crop and soil sciences. He stayed at Auburn and pursued his master's degree under the direction of Dr. Scott McElroy, focusing in molecular aspects of herbicide resistance and troublesome turf weed species. From there, he graduated and moved on to Virginia Tech, where he is set to complete his PhD in August of this year under the direction of Dr. Michael Flessner. He's done some really interesting work in an area of weed science that's been long in coming, and that's on seed impact mills, i.e. seed destructors associated with harvesting operations. Eli's CV, the award list is long, so I don't want to go through it all, but what I'd like to say is Eli has excelled in the weed science community. He has competed and come out as the top student in both the Northeastern Weed Science Contest and the North American Weed Science Contest, which is one of the most prestigious accomplishments in weed science here in the United States. He will also be giving a teaching seminar this afternoon at 1:45. This will be sort of a short 20-minute seminar designed specifically for the students. And I would welcome you all to come and attend that. Faculty are also invited to attend. That is primarily focused on students. And with that, we've asked Eli to come and give a presentation regarding his research, highlighting his skills, highlighting his vision for building a research program within aquatic weed science here at the University of Florida Center for Aquatic and Invasive Plants. With that, Eli, I'll be happy to turn it over to you. Thank you for coming.
Thank you, Dr. Enlow, for that introduction. So, I am Eli Russell, I'm a PhD candidate at Virginia Tech, and today I'll be talking about my seminar, which is a multi-strategy approach to weed management in aquatic systems in Florida. So Dr. Enlow gave you a little bit of the background where I came from, but we'll kind of walk back through that a little bit. So, here we go. So I am married. This is my wife, Jessie. She's a civil engineer. We've been married for about 4.5 years now. We don't have any kids, but we do have a child, my dog. Her name is Jenny. She's a beagle. She is fully grown. She's just very short. We don't really know why, but she likes to spend time at the river. We've got her life vest, which is great for this position. Since it's aquatic, she's ready for the water, whether she knows it or not, but she's got her vest and she's good to go. So I did grow up in Alabama, which is located, if you look at the state, right down there in South Alabama, or as the locals like to call it, LA or Lower Alabama. So very different from the LA in California. But if you ever go to the first thing you'll know is that they have done a lot of reworking and built some new signs. And so we have our town slogan is the City of Opportunity. And when you see this is just a lie. Just no, there is no opportunity there. All the opportunity is outside of those signs. So just keep on going. Outside of that, there's a lot of crazy people that live there that like to catch rattlesnakes. So the other thing we do in our city is we have an annual rattlesnake rodeo where people love to get together, try to catch the biggest rattlesnake, which sounds crazy to me. Then, then they throw them in a bucket and try to race them and see which one's fastest. Then we'll eat them eventually. So it's a great time full of interesting people. So if you're ever near around like anytime between February and April, because it changes all the time, feel free to stop by and just observe. That's all you got to do. So anyway, after I left I did attend Auburn University, where I got a bachelor's in crop and soil science. And it was during this time that I took a weed science course taught by Scott McElroy and truly just fell in love with the topic of weed science. I thought it was very interesting, specifically herbicide resistance. I just thought it was insane that a plant could develop resistance to a herbicide and then not be killed by it when it had previously been able to be controlled. And so that really pushed me to stay at Auburn, where I did a master's in turf weed science. focused on herbicide resistance in annual bluegrass and goosegrass, which are two problematic weed and turfgrass systems. And so I did a lot of molecular work looking, trying to figure out the target site mutations associated with resistance. Now, completely changing courses, I went to Virginia Tech for a PhD where I focused on applied cropweed science. So I spend most of my time out in the field. I swapped a very stable 8 to 5 in an air-conditioned lab for 6 to whenever we're done in the field, whether it's hot or cold, it doesn't really matter. But it's been very interesting. A lot of my work has centered around harvest weed seed control, which is the idea that every time a farmer enters that field with their combine to harvest their crops, whether that be wheat or soybeans or corn, any weeds that they fail to control during the year are also brought into that combine, and the combine spreads those seeds back out to the field. But we know that those weed seeds are in the combine, so can we target them for some kind of control before we throw them back out to the field? And so that is what my research has revolved around, which has led me to following lots of combines and collecting a lot of things coming out of the back, as you can see there on the right. Now, all of these experiences have really built a passion for weed science for me. I really, really love weed science. I love all the aspects of it. I've worked in different systems as well as different types of research with molecular and applied. Weed science even bleeds into my hobbies. As you can see, one thing I really love to do is take pictures with overly large weeds for their species. I'm trying to get my signature pose down for all my weeds. They pretty much all, in a way, have this pose now. But I just love weeds very much, and I love working with them to the point that it's not really working, it's just a fascination with them. And so because of that, in 2022, I applied for a travel enrichment scholarship through Southern Weed Science Society. And I actually got to come here to Gainesville and meet with Dr. Greg McDonald, as well as Dr. Farrell, and Dr. Enlow, and Dr. Sperry at the Center for Aquatic and Invasive Plants. And I learned all about aquatic weed science. And it was a very fascinating experience because that was something I'd never really considered as far as a career. I love weed science and I've enjoyed working in turf and crops, but I had never thought about aquatic weed science. And so when I went back home, I was so excited. I remember telling my wife, I've got to work in this. If an opportunity arises, I want to be there. And thankfully, here I am interviewing for a position, so fingers crossed. But as I told other people about this opportunity and talking about weed science and aquatics, they all kind of said the same thing because none of these people had any kind of knowledge about aquatic weed science. And one of the big questions that comes up is, why do we even need to control them? You know, it can't be that bad. And well, aquatic weeds really affect a lot of things. For instance, as far as public use, all of these waterways that have aquatic weeds in them can affect, how people use them, whether that's for boating or fishing or hunting, to name a few. But it also has an impact on commercial use, whether that's for shipping or power generation in some of these rivers, as well as potable water supply from springs. Aquatic weeds really affect all of these systems. So once you explain that we really need these waterways and these bodies of water free of weeds, the next question is, well, it can't be that hard to control these weeds. I have a turfgrass pathologist friend who loves to joke that there's only 3 weeds in aquatics, and once you know those 3, you got it checked off. But that's not the case. There are a ton of aquatic weeds, and it's not really that easy. And so when you look at really the numbers, especially here in the state of Florida, millions of dollars are being spent every year to control aquatic weeds. And so that means that we have a problem. There's a problem here. It's taking a lot of money to control it. And so, there needs to be research in this area. And then the final question I would get from everybody was, well, if you want to do this position, are you qualified? I'm coming from a turf background as well as a crop weed science background. And I know there's probably people in the audience who are looking at that and saying kind of the same question. Are you qualified for this position? And I'm here to hopefully change your mind today Because first and foremost, I am a weed scientist. The principles of weed science don't change regardless of the system you're in. And being able, now there are some nuances when you're in a new system, but understanding the principles of weed science is something that I feel like I have a very strong connection with because I've worked with it in different systems and I can see those nuances across this. I also have a very diverse background. I've worked with problematic weeds in turf, problematic weeds in row crops. I've done molecular weed science research looking at herbicide resistance. I've also done a lot of applied research looking at not only working with herbicides, but also working with mechanical weed control. So I have this large foundation to build upon. And as a weed scientist, and as I walk through this presentation, I'm going to kind of point out some of the issues I see in aquatics, how that connects to my background, and what kind of research I would like to build upon in that. So when we look at this chart here, you don't have to focus on each little individual herbicide, but each one of those little hexagons is a registered herbicide in the world. So we have the Herbicide Resistant Action Committee. They put together a mode of action classification chart for all their herbicides that really characterize how these active ingredients work in the plant. And so when you look at this, there are a ton of herbicides out there on the market that you can find in some capacity. But when we take a closer look at this, all of these pink dots represent herbicides that are available for aquatic use. And as you can see, there's not many dots up there. That there's not a whole lot of herbicides that are registered for aquatics. We're not even hitting all of the modes of action. There are entire modes of action that there are just no herbicides available. And so having a handful of herbicides isn't necessarily a problem. But the importance of that is good stewardship of those molecules. Because when you have so few, losing any to herbicide resistance can be devastating. Because we're not losing just one that we can just easily replace. And so fluoridone resistant hydrilla is kind of a case study for this in aquatic systems. Now I know this happened decades ago, but there's still research showing that these populations, even though fluoridone hasn't really been applied in some of these lakes, these populations are still there, meaning that the herbicide still can't be applied. And so being able to work around resistance and make sure that we don't have any resistance development is very important, because not only do we have fluoridone resistant hydrilla, there's been reports of diquat resistant duckweed, which duckweed isn't necessarily a problem, but you still don't want resistance. And then also I've been told that there's suspected resistance to indothal and hydrilla that could really be a problem, because indothal is really one of the useful products for controlling hydrilla. It's usually tank mixed with a lot of different products, and so having that product gone could be a big issue. And so pairing this with some of my research, during my master's, I got to participate in a lot of herbicide screens. So I worked with these group 3 herbicides, which are mitotic inhibiting herbicides, so they prevent cells from dividing properly. And they do this mostly by working in the root. So these herbicides you generally apply before the seed emerges. And then as the seed emerges, it comes in contact with that herbicide and it kills the weed. Now, when we're collecting resistant populations for this, it really set up a challenge, because we had these full-grown plants that we collected that we can't test for resistance because they're grown. We need seeds. So then you have to take these plants, you have to bring them to the greenhouse, you have to grow them out, then you have to collect seeds and you have to put them in a freezer and give them some cold storage. And then you have to plant them and spray them, and then 6 weeks later come back and rate. The next thing you know, this plant you collected, you don't know if it's resistant for 8-9 months. But what we found out is we can use one of these hydroponic systems where we just take a little bit of herbicide and spike this water solution in the bottom, and just take these full-grown plants and just pop them in there. And so if you look here on the picture on the right, we have four plants. The 2 on the left are non-treated. So what we can see is we have good root growth. It's great. The 2 on the right are treated. And what you can notice about these is the plant on the right has exceptional root growth, while the plant on the left has no root growth. All the roots that you see there are the roots that it started with. It didn't really put on any new roots. And so using this system, we're able to quickly call, in a matter of taking a process that took 8 months, we were able to shorten it down to about 4 to 6 weeks to being able to successfully determine whether a population was resistant to these herbicides or susceptible. Now, this wasn't the only step. So once we went through and we screened all these populations, which we ended up doing a couple hundred in both goosegrass and annual bluegrass, we needed to determine what the mechanism of resistance was. So this is a figure from one of the papers I was able to publish on annual bluegrass. And as we can see, we did have a mutation on the alpha tubulin gene in our 3 resistant populations that had been reported previously in literature to confer resistance. So we knew we had a mutation in these plants, but what does that mean for our stakeholders? Well, we took it another step and we started looking at cross resistance and doing dose response studies. So in this study, we have again our 3 resistant annual bluegrass populations. And so on the graph on the left is prodiamine, which is group 3 herbicide. And then on the graph on the right, we have dithiopyr, which is another group 3 herbicide, just in a different family. On the x-axis, we have our herbicide rate, and on the y-axis, we have our seedling emergence as a percent of the non-treated. And if you look at that, right around that 100 kilogram of AI per hectare mark, you can start to see a very clear separation between our 3 resistant populations and our susceptible population. Which is very concerning because we have 2 herbicides that are in the same group, but a single mutation has led to both of these popular herbicides being lost. And we did a similar study with goosegrass that had a different mutation on the alpha tubulin gene, and we found cross resistance to 4 group 3 herbicides. So all of a sudden we have 4 popular herbicides that are no longer able to be used for these golf course managers. And so what does that mean for aquatics? So when we go back to this graph and we look at our ALS, we have 4 groups, 4 herbicides in our ALS inhibitor herbicide mode of action. And it has been reported multiple times in terrestrial plants that there is a mutation on the ALS gene that can confer resistance to all herbicide families within ALS. So it is possible at any given moment that any of these aquatic plants we're managing to develop these mutations because they happen at random. And a mutation like that could result in 4 herbicides just directly gone. And that might not sound like a lot, but that's more than 20% of the herbicide options that are currently available for aquatic systems in Florida. And so when you think about one little mutation taking away 20% of your herbicide options, that can be detrimental to aquatic plant managers. So in this position, I would like to do some herbicide resistance research, because I think it's very important. And I think that there's an opportunity to kind of use some of these hydroponics systems or develop something new. Aquatic systems are very unique. And so there's a lot of opportunities for innovation in herbicide resistance screening. But I want to be very proactive about resistance screening because herbicides do make up a big part of what managers use to control aquatic weeds. And so anytime that there's even a thought that there might be resistant populations, making sure we're on top of that and confirming any resistance is very important. So a good proactive screening process, as well as cross-resistance testing. We need to ensure that we're not losing more than just one herbicide anytime there is resistance. And then also resistance mechanism detection. Whether that determining whether or not we have a mutation in a gene, or if we're developing resistance through other ways, such as metabolism of these herbicides, can really impact management. Because if we have metabolism-based resistance, we could start jumping these herbicide families, and instead of losing just ALS, we might lose some of our auxin herbicides as well, because metabolism is a very interesting kind of resistance, because who knows what herbicide it will start breaking down next. So understanding what resistance we might have across the state, as well as what mechanism is responsible for that. And then finally, using this information to determine what remaining control options are there. And while there's more than just herbicides, we're going to kind of take a step into some of those remaining control options is as far as herbicides are concerned, looking at herbicide efficacy trials. Now, at Virginia Tech, I had the opportunity to do many of these in corn, soybeans, and pasture grasses. And so I know what you're saying. Well, I don't see any water out there. That's true. But when it comes to herbicide efficacy trials, we're always looking for the same two things. Looking at our efficacy on problematic weeds, and we're looking at injury to non-target species. And so I have plenty of experience rating both of these. And our non-target species change, our problematic weeds change, but the principles at the root of it are all still the same. So with that in mind, I would like to do herbicide efficacy research. I think that there's a big opportunity to look at herbicide tank mixtures. I know Dr. Sperry and others are starting to look at some of this work by combining products for control, because this is a great way to manage resistance. We just spray one mode of action, we're really over and over, we're increasing our chance of resistance development. But if we can get 2, maybe 3 modes of action in that tank, we can really delay resistance development. But also by combining products, I believe there's an opportunity to start looking at, can we maximize our weed control efficacy on problematic weeds? Can we reduce exposure time? So some of these herbicides require long exposure time, and if we can combine multiple products, can we actually reduce the amount of time that we need to expose these weeds to herbicides, as well as look for any synergy between products. So can we get better control with two than we would expect with either one? And then finally, we need to make sure that we're not minimizing non-target target injury. So I believe looking at these tank mixture combinations in this herbicide research be a great way to accomplish these goals, and these are some of the areas that I want to look at. And also, I believe that there's a good chance of getting maybe some of this funded through company funds. I know in the aquatic space, company funding is not as strong as what I'm used to in as far as turf and row crops. But if we're doing work directly with some of their products, maybe there's an opportunity to get some funding. So it's an outlet that I'm willing to look into and hopefully be able to secure some. But herbicides are not our only control option. When we're looking at an integrated weed management system and combining multiple weed control methods to really get a more effective and complete control, there's a lot of other options that you can look at. And so I think that there's a great opportunity to dive specifically into mechanical weed control. Because one, this provides an option for removing or killing weeds that are problematic in these systems, even if they're herbicide resistant. Aquatic harvester doesn't really care if that weed is resistant to herbicides or not, it's going to collect it just the same. So I think that this is an option to look into. And if you, in my research, I found less than 5 relevant peer-reviewed articles surrounding mechanical weed control since 2000 in aquatic systems, which means there's not a lot of research being done there. You can find a lot of review papers, but very few people are actually doing research to determine if this is a good control method. We have data from the 70s and the 80s, but a lot has changed since then. Now, throughout my PhD, I've had the opportunity to work with mechanical weed control a lot. So that has come in the way of seed impact mills, as Dr. Enlow mentioned. So I've been able to work with 2 different companies, De Bruin, which makes the integrated Harrington seed destructor that you see on the left, and Redicop, which makes the Redicop seed control unit on the right. Now, these machines attach to the back of a farmer's combine, and they're powered by the combine. And instead of spreading those seeds back out to the field, they go into the mill, and the mill basically pulverizes those seeds. So we're returning non-viable seeds back out to the field. Now, these mills originated in Australia. And so that was designed pretty much for like dry land wheat. And so their systems are very, very different than what we would expect here in the US. Because one, we wanted to start using these in soybeans. Well, they're not using those in soybeans. It wasn't designed for soybeans. And then also our wheat here, especially on the East Coast, experiences a lot more rain during harvest season, which means there's just a lot more moisture in our crops. So it's different systems. So anytime you start working with any kind of new technology, the first thing you do is you start small. You never just go straight to the commercial use. You want to start small and see if it's viable. So we were able to secure stationary test stands from both companies. And what we could do is, instead of having to chase a 30-foot combine header across the field, collecting all this chaff, we could just take 1.5 kilograms chaff, which is about this much, put it on this conveyor belt. We could test a wide variety of seeds. We would run it through using a tractor PTO to power the mill, catch all this material, take it to the greenhouse, and evaluate any surviving seedlings. So this way we could determine, is this mill actually killing the seeds like it's supposed to? And since a lot of these seeds, especially for soybeans, they do not have in Australia, this is a very valid question. We need to make sure it's killing the seeds that we care about. But also, since this mill is connected to the combine, we need to see how it affects horsepower draw. And if in different harvest conditions, if it increases horsepower draw, that means more money for the farmer, because that's more energy used by the combine, more fuel, and therefore more money. So it was kind of a two-part process of, are we gonna kill the seeds? And how does different harvest conditions affect the power usage by the mill? And so, starting with some of the results from our seed kill, now there's a lot of data here, and I'm just going to kind of briefly describe what's going on. On the left, we have our soybean weeds. On the right, we have our wheat. In the maroon bars, we have our IHSD. In our orange, we have the Redicop Seed Control Unit. X-axis is all our species we tested. And then on the Y-axis is our seed kill. Now, one thing I want to point out, it looks like there are a lot of differences, but our seed kill for soybeans starts at 98%. So we're just in that top 2%. And for our wheat, we're starting at 90%. So in that top 10. And one thing I do want to point out is doing great. The impact mills and our stationary mills did a really good job of killing our seeds. We killed more than 98% of all the weeds we tested in soybeans and greater than 91% of all the weeds we tested in wheat. So it's very promising at reducing the amount of seeds that keep going back into the seed bank. But what about the power usage? Because some farmers like to push their combines a little harder than others. And since this is processing harvest residue, an increase in harvest residue should be an increase in horsepower draw. So we wanted to look at that and kind of round out full testing of this mill to determine how it would really work in the field. So for these graphs, we have the Redicop on the right, IHSD on the left. Our soybean chaff is in orange and our wheat is in maroon. Our horsepower is on the y-axis, and our chaff flow rate into the mill in kilograms per second is on the x. And as you can see, if a farmer wanted to really push their combine and really overload that mill, we get a very linear relationship in the amount of horsepower we use versus the amount of chaff going into that mill. So this is a great experiment to kind of help answer some of those questions, because we have a farmer that we work with that he likes to go real slow. So he's kind of on that 1 kilogram per second side, and he's not really using a whole lot of horsepower to run his mill. But then we have another that likes to push it closer to that, like 2 to 3. And so he's using a lot more horsepower and a lot more fuel than the other farm. So once you figure out all these little things, the next thing you do is you take it to the field. So we're able to put these mills on some commercial combines, and it resulted in us chasing combines all across the state and collecting all kinds of chaff. It's just great, just dirty work. But from this research, we again want to test two things. We want to test, are we killing those seeds still? Because that's still a question. We showed it works in stationary mills, but does it actually work when we get to the field? And then two, a combine actually has 2 residue streams coming out of the bag. Now, normally these combine, but in the case of an impact mill, we keep them separate, because that chaff fraction, which is generally your smaller residue, is what's processed by the mill, while the straw is not processed by the mill. So we wanted to make sure that the weeds are actually ending up in the chaff and not being lost in the straw fraction. So if your combine's tuned correctly, which is a big if when you're talking to farmers, we should expect most of those weeds to go to the straw or to the chaff, but we still needed to double check this, because this is all rounds out testing the full efficiency of this machine, because it doesn't matter if it's killing seeds if we're not getting seeds to that mill. So I'm only going to talk about results for one crop and with one of these mills, but the results were very similar across the board. So for the IHSD and soybeans, We'll start with our lost, percent of seeds lost in straw. So on the x-axis, we have the species tested. On the y-axis, we have percent of seeds that we found in straw. And the good news is this farmer was very good about tuning the combine, and we lost less than 5% of our seeds in straw, which is good. It means we're not just throwing them back out to the field, and they're actually making it to the mill. But what about those seeds that make it to the mill? This graph is set up the same as the other seed kill graphs. And as you can see, we're still killing greater than 98.5% of our seeds were tested, which is really good. So we've got effective control, or effective kill rates of these weeds. We're not losing very much to the straw. And we're able to kind of round out this project by being able to tell a full story on what this machine is capable of. And so how does that compare to some of the research I want to do in this position? Well, there is, again, this direct correlation when working with mechanical weed management, which weed control, which again is pretty different from working with herbicides in terms of scale and some of the metrics you're measuring. But I think that there's an opportunity to pair, especially with some of these smaller companies that design these, or these companies that design smaller harvesters like Aquarius Systems. I think that there's a great opportunity to look at kind of the rate of harvest. With aquatic weed harvesters, one thing that is a big problem is you can't really do a whole lot of acreage. So trying to figure out what we're actually capable of harvesting. Can we get 1 acre? Can we get 2 acres? Well, we have, does that change when we're talking about floating plants or submerged plants or by species? What are we capable of doing in a day? What about harvest efficacy? If we're talking about submerged plants like hydrilla, are we actually getting all those fragments into the harvester? Or are we just creating new infestations where these fragments are going to float down to another part of the lake or the river and start a new population? Or what about with submerged vegetation? Are we going to actually get all the way to the bottom and be able to bring all of those plants in? And especially when we're talking about hydrilla that has tubers, the more of that plant we can remove, the longer we can have before those tubers start putting new plants out. So kind of determining harvest efficacy with these machines. And then finally, I think that there's an opportunity to really take that data and figure out and optimize our harvester use based on weed density. So going out to a lake and saying we have this many weeds in this area, is it going to be better to use this harvester? Is it even economically feasible? Or do we need to do something else? So I think that as we have proven in the past, harvesters do have limitations, but I think that there is a way to basically figure out when it can be used and when it can be used effectively. Now, with that, kind of optimizing this harvester use in Florida for aquatic weed managers, most of them follow the idea of maintenance control. So what we want to do is we want to keep populations at a low level. But this requires a lot of coordinated and consistent management of these populations to make sure that they stay small, because we can't really eradicate these plants. So we just want to keep them at a low level so that they're not really affecting anything. But this can be very difficult because when we're talking about weeds like water hyacinth here, they can double in size every 7 to 14 days. So how can we go about actually tracking these densities and controlling them? Well, as a PhD student, I've had opportunity of working with some of these population density studies. So with that impact mill, we saw that we're killing the seeds. We're not losing a lot of combine, but is that translating to reduced weed pressure in that field? So we set up these massive field scale studies with common ragweed and soybeans and Italian ryegrass and wheat and barley. And we basically split these fields in half, harvested half of it with an impact mill, and didn't harvest the other half. And so this is some of the results from that experiment. So on the x-axis, we have our field. The y-axis, we have our common ragweed density. The orange bars is the on side, or the side that we harvested with the seed impact mill, while the off side, or the maroon, is the part we didn't harvest with the seed impact mill. And so we harvested in the fall, came back in the spring after planting before our post-emerge herbicides went out, and we counted the weeds that had emerged. And as you can see, we had a mixed bag. We had some that had reductions, some that really didn't in this first year. But if you've ever worked with a soil seed bank, you know that practices like this where you're reducing the amount of seeds going to the seed bank can take a long time before you start seeing consistent results because soil seed bank has very good buffering capacity for its weeds, which is why it's really hard to work with and get consistent results. But keeping in line with that, can we do something that over time slowly starts to reduce these populations to the point where they're almost not really there? I think that there's an opportunity to pair kind of with the Florida FWC. They have this great website on what's happening on my lake where they have all of this data that they have collected for years, looking at what weeds are out on these lakes, what control they have implemented, how many acres they've controlled of these weeds. And I think taking this historical data and pairing it with kind of some UAVs as well as remote sensing and machine learning, would be a great collaborative project because I don't have a whole lot of experience in this. I have experience working with, with weeds as far as a population scale, but remote sensing is not necessarily my strong suit. So collaboration would be really good for this project. But can we basically use this data and determine based on what control methods have been implemented and what standing densities we have in the years to come, to basically use UAVs to quickly scan a lake and take pictures and determine what density is out there of these specific weeds and basically optimize our control. So can we go out there with a drone, fly it and say, okay, this population has roughly this density on the lake and this biomass. How long before we really need to come out here and do something? Where it might, in a normal system, somebody might come out a week early and we could really have waited it and maximize our control, maximize our usage of our resources. So I think that there's an opportunity to really dive into kind of these large scale projects so that we can maximize the resources that the state has for controlling these weeds and keeping them out of the water bodies. Now, additionally, with some of the research that I've done at Virginia Tech, I wanted to continue some of that genetic research that I did during my master's. And so I set up this flowering time experiment where we were trying to determine a resistance mechanism to harvest weed seed control. So the idea of this is if a plant can flower earlier, can produce seed earlier, and shatter those seeds earlier. If those seeds fall off that plant or shatter before the combine makes it to the field, can't make it to the combine, they don't make it to the mill, and they survive. So what we did was we did a selective breeding where we crossed our earliest flowering plants, produced two different populations, and over 3 generations, we were able to shorten in the greenhouse our average time to flowering by 30 days for both populations, as well as reduce the variability within these populations, which is very concerning, very interesting to me, but for farmers, this is a little concerning. But understanding kind of the potential and the adaptability of these plants is very important because if we know that, for instance, some of these plants are flowering early, the next question here is, are we actually still getting the same seed production out of these plants? What about biomass accumulation? Are they actually, if they flower 30 days sooner, where are their resource allocations going to? And does that actually affect its ability to be a weed? And so understanding some of these weed biology questions is really important, I think, for truly being able to control a weed and knowing what its weak points are. So one thing that I really want to do in this position is also try to answer some of these basic questions about aquatic weeds. There is some research out there for the big ones, but there are also a lot of important aquatic weeds that we don't know a whole lot about. We don't really understand fully some of their growth rates, how often they grow and in what conditions, or their seed production potential, or their biomass accumulation. These are all things that could affect, especially biomass accumulation, affect the efficiency of aquatic harvesters. So understanding what these weeds are capable of in certain nutrient conditions as well as certain light conditions could really help influence management decisions about these weeds. So using all of this research, looking at weed biology, chemical weed control, mechanical weed control, I really want to create kind of a multi-strategy approach in this position for research, especially in weed management. Can we take chemical control that I talked about in terms of tank mixtures, pairing that with aquatic harvesters. And then maybe even incorporating some physical control methods or biological control methods where necessary, or applicable really for a lot of these methods to basically get better weed control. And we also want to look at 3 aspects for impacts for stakeholders, because a lot of my research is very stakeholder focused, because those are the end users a lot of this research. So can we, by combining these systems, increase our weed control efficacy, reduce our labor requirements, and minimize our costs in a lot of these systems? Now, with all of this research, you got to have some way to fund it. And after talking with Dr. Farrell and Dr. Enlow and Dr. Sperry, I believe that there's funding opportunities, external funding opportunities with the Army Corps of Engineers, Aquatic Plant Management Society, as well as Florida FWC and water management districts within the Florida Department of Environmental Protection, as well as I think, especially as a early faculty, or new faculty, that there are internal grants to compete within UF that I believe would be a good starting point for this position. Now, as a graduate student, I have always loved kind of the dynamic of the labs I've been in. I've always been in a lab of around 2 to 3 students, and that's kind of my 5-year goal in this position. I want to create a lab that's around 2 to 3 grad students with a technician. I think this kind of framework would allow me to get the research that I want done, but also be an effective mentor without having too much on my plate. I think after talking with Dr. Enlow, Dr. Farrell, I think that within 5 years, $250,000 a year in funding is a very reasonable number. I think I could potentially get higher, but I think that that is a baseline number that I feel like I can comfortably hit with the funding opportunities in this position. And then finally, outside of some of this research that I talked about would be individual, but I really want to do a lot of collaborative projects as well. I think collaborative projects really help answer some of those big overarching questions that take into account multiple areas of research that some of our managers are looking for. And so I've really enjoyed working on collaborative projects during my PhD, and that's something I really, really want to work on in this position. So with that, I'll take another opportunity to show more weeds. Regardless, here on the right, I was actually spraying some herbicide resistant plants. It doesn't stop. When you find a good plant, you just pause whatever you're doing and take some pictures. Dr. Farrell actually took that one in the middle. So back when I was here for my visit. So if you have any questions, I'll open the floor.
So let's open it up for questions. We can either start with the live audience here on Zoom. Let's go ahead and start with the live audience and raise your hand on Zoom. If you don't mind, we'll get that set up to start looking for questions from Zoom. Questions from the audience.
Yes.
I actually have 2, but I'll ask one first.
All right. Go for it.
So the actual mechanism for that resistance, that the alpha tubule mechanism for resistance in terms of plants, what is it?
So it's a target site mutation. So you get a nucleotide substitution that leads to an amino acid change that alters the protein, how it interacts with the herbicide. So typically with target site mutations that alter the protein structure, you usually get a little loss in fitness of the plant, but that little bit of loss of fitness is usually tolerated because now you can survive a herbicide application.
But the that protein is involved in actually the mitosis process.
Yes.
So when the herbicide comes into susceptible varieties, or not varieties, but weed species, it's somehow disrupting, or what is it doing that the others can't?
Right. So with microtubules, so it's a protein dimer of alpha and beta tubule. So those need to come together to form our microtubules. And with our group 3 herbicides, it really prevents that dimer from forming, and therefore microtubules from forming. So during mitosis, all the chromosomes line up. They don't have anything to actually pull them apart. So you get these like swollen cells that just not have double DNA. But with these mutations, a herbicide can no longer, that mutation falls within where that herbicide would bind to that protein. And it's off just enough that the herbicide no longer actually fits there, but the protein dimer can still actually bind and form and create microtubules.
And why, you said, it looked like there was a dosage effect, like when you increase the amount? Was I misunderstanding
Yeah,
that?
no, so we can eventually overcome some of that with enough herbicide, but from that study, based on kind of our like I-90 values, we saw that you would need about 4 to 5 times the legal labeled rate for those herbicides, if I'm remembering correctly, to be able to actually control those populations. So when you're talking about, you know, the label, the herbicide label is the law, you can't really spray that. So even though you can eventually kill it with a herbicide, it's not completely immune. It is outside of what you're capable of applying as a manager.
Patrick?
So all your research projects,
Yeah.
which one was your favorite? Why?
Which one was my favorite?
Yes.
Well, that is a great question. So I really think working with some of the stationary mill has been my favorite because it kind of it put me in the greenhouse, put me in the field. It kept me at home, so I was 6 hours away from home, chasing the combine. I could really do it on my own time. But it was also very interesting because we were able to test a wide variety of species and try to see if there was a difference between species. And we looked at 2 different, I only showed results for the chaff flow rate, but we also looked at chaff moisture. And I also think that, and how that affects our seed kill and horsepower draw. So we did that study both in soybeans and wheat as well, which was interesting. So it really was able to encompass a lot of the system that these mills would be. So that was probably my favorite, I guess, if I had to put a favorite on it. I just really love weed science research. They all have great things that I love. They all have things I didn't really love, but just enjoy doing research. So that would have to be my favorite. It's only slightly.
Okay, we have 2 questions from the audience. Emma, go ahead, please.
Hi, thanks for the wonderful presentation. You mentioned towards the end that collaborative research was a high priority for you, especially in your first 5 years. And I noticed that a lot of your photo citations went to Grow, and I know your advisor is involved in both Grow and Better Together. Could you tell me a little bit about your vision for what types of collaborative teams you'd be interested in joining or leading during those first 5 years?
Yeah, so I think some of the collaborations I would want to be in definitely would involve faculty at the Center for Aquatic and Invasive Plants. I think a lot of the research would connect. And so any way I can get involved with faculty there with the research would be important to me. Also, I think collaborating with Ben Sperry and the Army Corps of Engineers would be really fruitful because I know a lot of the research that I would like to do would be kind of picking up some of the research that they have looked at observationally as well as more in depth. So I think that there's a good collaboration there with the Army Corps of Engineers. Some of this research that would involve, you know, remote sensing and machine learning, I think working, I know University of Florida has just got a new center for artificial intelligence and kind of looking into that. And I think kind of collaborating with members of that group would be very beneficial for some of these projects. To just, I guess, name a few off the top of my head, is kind of some of that space I want to move in. And eventually, I think kind of capitalizing on some of those points I had about minimizing costs. I think trying to pair some of this research with economists would be really important, because if we're talking about figuring out all the metrics and, you know, what different weed control strategies cost, both in terms of the easy dollars you can see, but some of those long-term benefits of, you know, reduced populations and less time you have to go out to the field. I think pairing with an economist would make kind of that research much better. So, and then also pairing with ecologists. A lot of our aquatic work. We have one or two species we really want to kill, but there's a lot of native species out there that we don't need to kill. So figuring out what can we do as far as keeping those around, or if those are lost, what can we do in terms of restoration in those areas? I think that especially in aquatic habitats, those two fields kind of aquatic weed control and ecological restoration kind of need to go hand in hand.
Yeah, thanks for sharing.
Luke,
Yeah, hi. I was curious about your research on the mill and how it was damaging the weed seeds, and I was wondering how you calculated percent viability of the seed coming out. Like, did you have a separate collection method prior to the mill, or how did you do that? And then the second part is how that then connects to your population level analysis, because even 2% viability of weed seeds may not impact the population size in the field. And I know you did some of the research on population size, but I was wondering how you're trying to link those two different measures.
Yeah, so those are great questions. So kind of first starting with how we determine seed kill. So some research out of Australia showed that if you basically put a seed in the mill and it would be non-viable. So they pulled out all these seeds and they did tetrazolium tests, and they showed that even if a seed comes out whole, it is generally rendered non-viable. So we didn't feel like we had to necessarily try to search for every seed in our chaff that we were processing, because a lot of times we're talking about Palmer amaranth seeds, which is, you've ever seen those, I can hold about 1 million of those in my hands, and the mill just like pulverizes them into dust most of the time. So trying to find those in the chaff would have been a nightmare. So what we did instead was we started with clean fields, and we basically poured a known amount of weed seeds directly into the mill during harvest. So we knew how many seeds went in, and we caught everything coming out, and we took that back to the greenhouse and we did germination assays. So we planted them in trays and we counted them for 12 weeks. And using that number that came up, and we also in that same system put check trays out that had 100 seeds of each species, because we didn't want to say put weed species out there that had 20% germination rate. If that was the case and we had say 100 seeds and we only found one, we would suspect a 99% kill rate. But if you factor in the fact that you had 20% germination, 80 of those seeds weren't going to come up anyway. So now we're at a 95% germination rate. So we use these check trays in combination with what came up to determine our germination rate in this environment for those species, and then calculating based on what came up using that germination rate and what we fed to the field to determine our kill rate. So for the second question, as far as pairing weed density, you're right. When you look at a Palmer amaranth plant there have been plant cases where a single plant has produced 1.2 million seeds. It's the most they've ever found on a plant. So when you're talking about a single plant where you could have, thousands to hundreds of thousands to an acre, where each one of them can produce anywhere from 100 to 100,000 to 1 million seeds, killing 98% of those, that really, like you said, leaves a lot of seeds that could be returned back into the seed bank. But with that, there has been some modeling research done by, I believe it was Lavreet Shergill at Montana State. And he showed that as long as we're reducing our returns to the seed bank by at least 60% with and continue to use herbicides and other weed control methods, we can start to actually reduce that soil seed bank still, even with 40% of the weed surviving from the mill. We can still reduce that soil seed bank to by a noticeable level, in 8 to 10 years. So it just really takes time. Even though we are returning a lot of seeds back, if you add in other control methods such as still using herbicides, you can confound on that and overall reduce the soil seed bank.
Cool, thanks.
Yep.
okay, one more question. Oh, we got let's see, so. Go ahead. You're gonna be in the student session this afternoon. You can hold that question if you don't mind. Okay, thanks, Max.
Yeah, it's a very good question. You have a great presentation, and I was a little curious about the experiment that you mentioned where you consider soybean, wheat. Did you ever consider the genetics of those varieties? Because I'm assuming that you're trying to deal with, how to say it, the resistance or against resistance of these weeds, also the genetics of these genotypes play a role, because at some point these two genomes of the different species might be interacting. I'm just bringing the point because I think there might be some room, opportunities to deal or to expand a little beyond that, because here, I mean, I collaborate with breeders, and it's funny to see the discussions where they say, oh, the main factor for improving productivity is genetics, and then you see the physiology, no, it's management. So like things are interaction between those things. That's why I said probably in those experiments you use only one specific variety or use different sort of varieties of the varieties.
Yeah, so I guess in terms of soybean variety, where the genetics are, I don't think necessarily play a role for this research, because the mill only impacts wheat seeds at the very end. So the soybeans have already been separated out in the combine. They in no way get close to going through the mill. But as far as like weed genetics, there is something to be said about, do individual populations with small changes in their genetic makeup actually change our kill rate? So I know, so Nick Berry, who is the founder, we were not able to work with this company, but Seed Terminator is another company that makes impact mills. And Nick Berry, who is the founder of that company, has done a lot of research, and he started to dive into, are there small genomic changes that could impact seed kill? So he's looking at whether or not, if you have like a diploid ryegrass versus a tetraploid ryegrass, does that actually affect seed kill in terms of, and also weed seed size within those. So every weed that produces seed, some are going to be a little smaller, some are going to be a little bigger. So if we separate out size, does that also play a role in seed kill? So far, the data kind of says numerically, maybe, but statistically, the numbers are so close, you can't really separate.
Yeah, because I was a little curious with this thing that you mentioned about the mutation. So you have an idea what the mutation rate or the probability of a mutation to occur?
Yeah, so I guess the answer is always maybe. We think that there should be enough surviving ones that it won't happen as quick as what we've shown. I know another study in Australia did something very similar to wild radish, and they kind of saw the same things. They were able to halve the flowering time in 4 or 5 generations. But they've been using some of these techniques for decades now. So some harvest weed seed control has been done in Australia on some farms for nearly 40 years at this point. And while they're slowly seeing some resistance mechanisms potentially, it's been very slow because there's enough genetic diversity still out there that it's not solely selecting for some of those plants, especially when you factor in the seed bank. So those ones that shatter early, they're not always the ones that come up the next year. So we've kind of had a good buffer in terms of genetics. So there's been some observations that kind of point to that. But so far, as far as what's actually been happening in the field, we haven't really seen it as drastic as what we've been able to do in some of our selective breeding research.
All right, so we'd like to make our speech one more time. This concludes the seminar.